Bienvenue à cette soirée du vendredi, cette soirée de clôture du Colloque Wright pour la science. C'est le tout dernier soir de cette 21e édition. Merci d'être venus aussi nombreux à l'Université de Genève. Merci également à toutes les personnes qui nous suivent en direct sur le live sur Internet. Vous me connaissez, si vous êtes présents depuis le début de la semaine, je suis journaliste scientifique et mon nom est Sarah Servondada. J'ai l'immense plaisir d'être avec vous toute cette semaine et encore ce soir pour accompagner et animer les discussions. Et vous connaissez maintenant le principe. Le principe, chaque soir, nous accueillons un ou une scientifique de haut niveau pour nous accompagner dans ce grand voyage à travers les symétries de l'univers. Et ce soir sera un petit peu différent. Ce dernier soir va nous ramener à une échelle un peu plus humaine, puisqu'il sera question de symétries qui guident le développement du vivant, les symétries en biologie. Voilà, c'est un champ qui va nous emmener du côté des symétries des fleurs et peut-être même des motifs des guépards. C'est un voyage qui va nous faire passer des atomes aux orchidées. Et sur ce sujet, nous aurons l'immense plaisir d'accueillir Enrico Coen. Il est professeur au département de cellules et biologie au John Innes Center à Norwich, au Royaume-Uni. Avant de l'accueillir et de l'écouter, quelques petits rappels pratiques sur le déroulé de la soirée. Elle va se dérouler en 3 parties ce soir. D'abord, l'exposé scientifique du professeur Coen, qui va durer environ 40 minutes et qui sera donné en anglais. Si vous avez pris un petit casque à l'entrée, il pourra être traduit en direct par nos interprètes. Puis un panel de questions-réponses où le professeur Coen sera accompagné de 2 autres conférenciers des autres soirs que je vous présenterai tout à l'heure. Là aussi, la discussion va se dérouler en anglais et vous pourrez compter sur les petits casques audio pour les traduire dans la langue de votre choix, en l'occurrence le français si vous en avez besoin. Et je vous rappelle d'ailleurs que s'il vous plaît, ne réglez pas le volume de vos casques trop fort pour ne pas gêner vos voisins dans la salle. Un grand merci. Et 3e partie de la soirée, nous conclurons par un petit mot de conclusion par le professeur Thierry Courvoisier, président de la Fondation Wright. Encore un petit rappel pratique, c'est comment faire pour poser ces questions. Vous pouvez passer par deux moyens. Le premier, c'est la plateforme en ligne dont le QR code s'affiche juste derrière moi, plateforme Slido. Voilà, il suffit de prendre le QR code et vous pouvez poser vos questions sans attendre pendant la conférence. Le deuxième moyen, c'est tout simplement de nous faire signe pour solliciter l'un des micros qui va circuler dans la salle pendant la partie de questions-réponses. Et évidemment, s'il vous plaît, merci de bien vouloir mettre vos téléphones en silencieux par respect pour nos conférenciers et les personnes qui suivent ce colloque. Avant d'inviter sur scène notre orateur de ce soir, je souhaiterais donner la parole au professeur Yvan Rodriguez du département génétique et évolution de l'Université de Genève. Il est également membre du comité scientifique de la Fondation Wright. Bienvenue sur la scène, professeur Rodriguez.
À l'instar d'éditions précédentes, le thème des symétries de ce colloque Wright se prête à une articulation complexe et donc à un petit effort cognitif. Parce que les symétries, elles sont visibles dans les formules mathématiques, dans les lois de la physique, dans certains concepts, dans certains processus, mais aussi dans les choses que l'on observe avec les yeux. Autour de ce grand thème des symétries, nous avons exploré cette semaine, avec les professeurs Usan, Baumann, Amis et Copeland, le monde de la physique, ou plutôt les mondes de la physique, abordant le temps, les forces de l'univers, l'antimatière. Et pour ce colloque parlant de symétrie, il aurait été dommage de faire l'impasse sur une expérience que nous vivons tous ici au quotidien. Je fais référence aux constructions humaines, pourtant formidablement symétriques, comme le Parthénon. Alors, je ne fais pas référence aux constructions humaines, pourtant considérablement symétriques, comme le Parthénon ou la cathédrale de Genève, ou simplement ce pupitre peu stable. Je pense que ce sera pour une autre fois. Ce à quoi je pense, ce sont les symétries du vivant, les symétries que nous offre le vivant qui nous entoure. Nous tous ici, mammifères, en l'occurrence grand singe, présent dans cet auditoire, exhibons une symétrie bilatérale que d'ailleurs nous apprécions particulièrement. D'autres formes de vie, au fond des océans ou ailleurs, peuvent être organisées de la même façon, ou d'autres, différemment, comme les étoiles de mer ou les méduses, dont la symétrie, dont l'arrangement n'est, lui, pas bilatéral, mais radial. En réalité, en parlant de la vie, si on s'intéresse à la masse qu'elle représente sur la planète, ce serait plutôt aux végétaux qu'il faudrait tout d'abord s'intéresser, puisqu'ils représentent l'énorme majorité de la biomasse planétaire, loin, loin devant les animaux et même loin devant les bactéries. Et c'est justement des plantes dont va nous parler notre intervenant, le professeur Enrico Coen. Pour faire court, très court, après un doctorat à l'université de Cambridge, le docteur Coen rejoint, un peu plus à l'est de l'Angleterre, le fameux John Innes Center, où il s'intéresse aux fleurs et en particulier aux gènes impliqués dans leur construction, ceux qui agissent sur les différents axes de ces fleurs. Ces travaux sont remarquables, ils sont remarqués, ils lui valent la reconnaissance de ses pairs, reflétée entre autres distinctions par sa nomination en tant que membre de la Royal Society et aussi de l'US National Academy of Sciences. Parallèlement à ses activités de chercheur, Enrico Coen s'implique également dans la communication scientifique. D'un côté, c'est assez rare pour le faire remarquer, à la communication scientifique au sein du corpus des chercheurs. Il a entre autres publié une longue série d'articles dans la revue professionnelle Developmental, des articles dans lesquels il revêt l'habit et adopte le verbe d'une espèce ou d'une autre, parfois même d'une cellule, ou même d'un objet de la vie d'un chercheur, offrant un angle assez inhabituel de la science, original et même parfois assez poétique. D'ailleurs, c'est dans cet exercice qu'il a déjà eu en lien avec notre ville, avec la cité de Calvin, en discutant des activités d'Abraham Tremblay, un des naturalistes genevois du XVIIIe siècle, dont le nom résonne encore aujourd'hui au-delà de la République. Enrico, naturellement, s'intéresse aussi aux liens avec le grand public, à travers nombre de livres et d'articles, et aujourd'hui, à travers cette conférence. Aujourd'hui, donc, il va nous parler des symétries dans le monde du vivant, il va nous parler en particulier des plantes, il va nous parler de l'origine de ces symétries et des différents niveaux auxquels elles sont observables, et peut-être aussi, je l'espère, il discutera du rôle, en dehors de celui d'être apprécié, fortement apprécié esthétiquement par notre espèce, on peut s'en rendre compte avec ces arrangements, que certaines de ces symétries peuvent jouer dans le vivant et combien la sélection naturelle a agi dans l'histoire de la vie et agit toujours sur ces symétries. Enrico, the floor is yours.
Thank you very much. Can you all hear me at the back? Yeah? C'est bon? Right. It's normal to begin, when a speaker speaks, to thank the organizers for inviting them to give the talk. And this is, as always, I'm doing this, but I really want to thank the organizers. And to explain my great gratitude, I'm going to give you an illustration of something that happened to me today that has never happened to me at any other meeting that I've been to, and I've been to a lot of meetings during my life. What happened to me today was that I was thrown out of breakfast, okay? Now, why was I thrown out of breakfast? Well, I was having breakfast and having a nice discussion with my physicist friends here, Yasmin, Ed, and Jean-Pierre, and we lost track of time. Okay, we were just so excited and involved in our discussion. We lost track of time, and eventually the the waiters in the restaurant started to appear, and they were looking at their watch, saying, you know, it's finished, you know, you need to go because there's nobody else, we need to prepare the tables and so forth. So they moved us away, threw us out at breakfast, and then we carried on our discussion in the lobby. And actually, I was not only thrown out of breakfast today, I was thrown out of breakfast yesterday as well. The only difference is yesterday there were fewer waiters, because I think today they realized that this might happen, so they got more waiters together to throw us out. And you have to realize that I'm a biologist, and physicists and biologists are often separated. They often don't talk to each other. We're in different communities. It's like two types of children that never are allowed to play with each other. And, you know, I'm told, well, you don't want to play with these strange children. You know, they talk in a strange language, a mathematical language. You won't understand it anyway, and they're very weird, so don't bother. Just talk to your own community. And I think one of the great things of this meeting is that it brings together these communities, and it's been very exciting for me to hear different perspectives, very stimulating. So I think there should be more meetings like this, rather than fewer. So what I'd like to do today is try and convey some relationship between the rules of physics that we've been hearing about, and symmetries in physics, and the physical world with the biological world. And I'd like to begin by you imagining that you're going for a walk in the countryside around Lake Geneva, shown here. And if you look at the landscape, you're not struck, there's no striking symmetries, all right? The mountains are quite jagged, they're sort of almost chaotic. There's no obvious symmetry in this landscape. But now if we go to the lake, and we zoom into the lake, and we zoom very carefully into the individual molecules of the lake, then we do see a symmetry. We see that the water molecules inside the lake have this bilateral symmetry that you just heard about, this plane of symmetry. Remember from the previous talks, the notion of symmetry means that there is invariance to a certain type of transformation. So if you reflect the water molecule through its middle, then you end up with the same looking molecule. All right, so another way of thinking about it is in terms of repetition. The left half of the molecule repeats, and in mirror image form, the right half of the molecule. So why is water symmetrical? One of the things we've heard today is this symmetry is itself an expression of another symmetry, a fundamental symmetry in physics, which is that the laws of physics are invariant to translation in space and time. That is, the laws of physics over here are the same as the laws of physics over there. Now that might seem like an obvious thing. Why wouldn't that be? But there's no logical reason to expect that. It's an amazing thing. The laws of physics do not change in space and time, and that is an incredible symmetry. It's a symmetry because it's a lack of change following a transformation that underlies the whole of the universe. And it also underlies the symmetry of this molecule. If the laws of physics were different for different atoms, you wouldn't have this symmetry because the hydrogen on the left would obey a different rule from the hydrogen on the right. We wouldn't even have hydrogens. It's the fact that this law of physics is obeyed in this symmetry-preserving way that it means, as we've heard, that we have the rules of quantum mechanics, that we have a limited number of elements, and that these elements combine in a limited number of ways. So this symmetry is itself an expression of the fact that every atom follows the same rules of quantum mechanics. Now this molecule, you'll see the scale, is 10 to the minus 10 meters. So the human scale is about 1 meter. That's the scale at which we live. This is 10 orders of magnitude smaller. It's about 10 billionth of the size of the world in which we live. Now you also find symmetries, water adopts symmetries at slightly larger scales. So here, for example, if we were to look at the lake on a rainy day and zoom into some water droplets, we see that they also exhibit approximate symmetry. There's something like 10 to the 21 water molecules in a water droplet. Why are they symmetrical? Because every molecule in that water droplet is following the same rules. And one of those rules is water molecules adhere or interact more strongly with other water molecules than they do with air molecules. And because this behavior is averaged out over all the water molecules, and they're all doing the same thing, you end up with this spherical symmetry. So we see again, it's a reflection of this fundamental symmetry in physics that everything is following the same set of rules. Now, if you go up into the mountains and you look at the snow, you also will find that water adopts another type of symmetry. The snowflake, for example, has hexagonal symmetry. So why does a snowflake have hexagonal symmetry? Well, that's because when water crystallizes, the water molecules crystallize, they form this kind of hexagonal arrangement, and that hexagonal arrangement, which has 6 planes of symmetry, propagates through as the crystal grows, and because every branch in our growing crystal snowflake obeys the same rules, we end up with a molecule with hexagonal symmetry. So we see that in the world of physics, we find symmetries at the microscopic scale because of these conserved behaviors or conserved rules. Now, if you take a rocket in Geneva and you then you boost out of space to have another look, you will encounter another symmetry, which is the symmetry of the Earth. So now we've gone way beyond the human scale, much larger than the human scale, 6 orders of magnitude, 6 or 7 orders of magnitude, greater than human scale. Why does the Earth essentially have almost spherical symmetry? It's the same thing. It's because all matter follows the same rules of gravity. So we see it's the repeated application of the same often simple rules that lead to these symmetries. Now let's go back to our walk around Lake Geneva, and if you're very lucky, what you might see is this. Now here's another type of symmetry, or it's a bilateral symmetry, a single plane of symmetry in a slipper orchid, all right, which I believe grows in the Alps. And the question, and this is now much more at our scale, all right, this is only 10 to the minus 1 order of magnitude, and we can see it with our naked eye. And now we look at this symmetry, and we think, what is the relationship between this symmetry and the other symmetries that we're seeing either in the microscopic scale or the macroscopic scale or the astronomic scale in physics? Is there a relation between them? Is there something totally different? This is a living thing. Are the rules for symmetry here different? What's going on? And that's the problem I want to discuss with you today. But we're not going to look at orchids, because orchids are not very convenient for answering this question, for doing scientific experiments. We're going to look at a different species, which is the snapdragon. Antirrhinum is the Latin name. I think in French it's either you call it muflier or gueule du loup. So it has this same plane of symmetry, but it has the advantage that we can analyze particularly the genes, the genetic control of the development of this flower. Now, I've spent 40 years of my life studying this flower, okay? So you might think that's a very sad way to spend your life studying one type of flower, how boring. But I have to say, I've enjoyed every minute, well, most minutes, and it's been a journey, scientific journey, a fascinating scientific journey. And I want to try and explain some of the principles that I've gleaned through that scientific journey over the years. Okay, so one of the interesting things about this, I keep pointing there, but you're looking there. One of the interesting things about this flower is that there are mutations that change the symmetry of the flower. So here, for example, you can see a symmetrical flower that's got 5 planes of symmetry running through it. And it's the same species. It's the same species as the normal antirrhinum or normal snapdragon. It's just had a mutation. It's had a change in one of its genes. Okay? And that change has fundamentally changed the symmetry of the flower. Okay? So what is a mutation? What is a change in a gene? Well, if you remember, genes are actually encoded in the DNA, this long double helical molecule. All right. So DNA is a series of what are called bases, sequence of bases, and these are stacked up, and they're stacked up in such a way that they have this nice double helical symmetry. So DNA is also exhibiting symmetry due to the way these molecules interact with each other. And you'll see it also, this symmetry reflects repetition, the fact that the bases are repeated in a long sequence. That's what allows you to see this symmetry, because you essentially can move up by one and twist, and you'll end up with an identical looking molecule, all right? Now, the snapdragon, all our genes are encoded by DNA. Your DNA is no different from a snapdragon's DNA. The snapdragon's DNA, there's about 500 million of these bases stacked up, all right? One gene is a small stretch of this DNA, of, say, a few thousand bases. And a mutation is often just a change in one or two bases. Just one base can be sufficient to have a mutation that changes the sort of properties of the DNA and how it influences the development of the plant. And that's a remarkable thing. So you just change a few bases. Remember, we're dealing with a molecule of around a few atoms. We're talking 10 to the minus 9 meters. So 9 orders of magnitude smaller than us. And that change propagates through and influences the symmetry of something that's 7 orders of magnitude larger. Now, that is very different from the world of physics. So if you take a few atoms in a water droplet, it doesn't change the shape of a water droplet. And also, if you think of the hexagonal symmetry of our snowflake, the snowflake has a hexagonal symmetry which is reflected in the hexagonal symmetry of the crystallizing water molecules. But here, the symmetry of the flower has no relationship to the helical symmetry of the DNA. So here's the question: how does changing a few bases of a DNA molecule with helical symmetry lead to a change at a much higher scale in the shape of this flower? That's the question we want to address. Now, addressing that problem is quite complicated in the first instance, because the flower is quite a complicated thing. So what I want to do is first begin with something simpler. Let's begin with a single cell. And plants are particularly interesting for studying this problem of how you go from one scale to the next, because plant cells, unlike animal cells can move around and slide past each other. Well, plant cells can't. They have these walls around them, cell walls around them, that locks the cells into position. That means they can't move, but that means that we can understand the process much more easily in a plant than we can in an animal. So what I'm going to, by studying a plant, we actually provide a way of understanding the principles behind this, but these principles are going to be applicable to humans, to animals, and so forth. It's just that here we have, in a sense, a simplified system. We don't need to worry about cells rearranging and moving past each other. So let's begin with a single plant cell. Let's first take away its wall, remove the wall, and we're left with what's called a protoplast. So here's a single plant cell, a protoplast. It's essentially got a membrane around it. It has spherical symmetry. Why? Because it's got an internal pressure. It's got an internal pressure that's pushing out, and pressure acts equally in all directions. All right, this pressure is called turgor pressure or osmotic pressure, and the pressure is resisted by a membrane all the way around the cell. And that's why, again, we're seeing the same rules, simple rules of physics. We just have a pressurized container, and that is giving us this spherical symmetry. And the question I want to ask is this: if you were to leave this spherical cell to grow and make its wall, so it starts to make a new wall around it, and it starts to grow, what can happen is that it grows into a cylindrical structure. It changes its symmetry, okay? So we start off with spherical symmetry, which essentially has an infinite number of planes of symmetry in every orientation. And now we go to a cylindrical symmetry, which has a more restricted set of planes. So we've changed the symmetry. The cylindrical symmetry has a single axis of rotation about which you can rotate, whereas a sphere has an infinite number of axes about which you can rotate and keep the shape. So how do we generate cylindrical symmetry from spherical symmetry? That's the thing I want to try and address. And to do that, I think, I'm going to give you an experiment. Okay, so, right. It's the first time I've done this experiment in, in this kind of environment. So can you all see this? Yes, so here's a balloon. I'm blowing up the balloon like a cell, and it's sort of adopting spherical symmetry. All right, so that's the norm. The question is, how can I make this balloon adopt a different type of symmetry, turn it into a cylinder? So what I'm going to do is, it's leaking a bit, I think, here. Never mind. I'm going to wrap some tape around the balloon. Nice sound effects. Oops, I've twisted it now. Yep. Few experimental modifications are needed here. Right, I'm going to cut this now. So what happens now when I inflate the balloon? Okay, that's the question. So, is that cylindrical? Kind of cylindrical symmetry. I made a mess at this end, but I hope you can see that it does the job, right? So what I did was I basically generated this change in symmetry by wrapping the cell with a constraining set of fibers around it. Okay, but there's no hand in my plant cell. This has to be, the plant cell has to do this on its own. So the question is, how does the plant cell do this? All right, so if we go back to the presentation, back to the presentation. So get rid of me, yeah. Right, now a plant cell, the protoplast, has fibers around it, just like our balloon, and these fibers are cellulose molecules. So cellulose molecules also have symmetry. They have translational symmetry, because these cellulose molecules are made of glucose subunits, and these subunits are joined together to form a long chain, and that's what cellulose is. And they have translational symmetry because it's just a series of units, one next to the other. So if you shift along by one, you end up with the same molecule. So they're wrapped around, and the question is then, how are we going to control these cellulose molecules, all right, to change the symmetry of the plant? So we need to understand how cellulose molecules are made. Now, I mentioned DNA. DNA is copied to explain how cellulose is made. You imagine the DNA is copied into another molecule which has a single strand, which is called RNA, which has the same sequence of a basis as the DNA. And then the RNA molecule, the stretch of RNA, is then copied into protein. A protein is often an enzyme that can catalyze or cause a certain reaction to happen. So there are proteins, for example, that promote the formation of cellulose, that assemble the glucose units to make cellulose. You'll see here we're already dealing with a process of amplification. One DNA molecule can make many RNA molecules. Each RNA molecule can make many proteins. So we're starting to see how we can move up the scale by amplifying things, all right? Now, the cellulose protein, the protein that makes cellulose, is itself guided by another protein that I'm going to need to introduce, another type of protein, which is called a microtubule. Now, microtubules are made of protein subunits that are joined together to form a helical tube. All right, and this helical tube grows at one end. As you add more units to one end, this helical tube grows. All right, and to show, I think I've got a movie showing that. So here's a movie where we're staining the microtubules in a plant cell. And you'll see, I hope you can see, that the microtubules are moving around because they're growing at one end. Each is growing at its particular end. So these are microtubules. Now, these microtubules guide the synthesis of cellulose. To explain how that works, I'm going to use a sort of analogy here. The microtubule is like a railway track. All right? On the railway track is another protein, the protein that synthesizes cellulose. That's called cellulose synthase. So that protein moves along the railway track, and it spins out cellulose, like smoke, all right? So in that way, the direction of the cellulose depends on the microtubule, because it's the microtubule that's guiding, all right, the cellulose synthase that makes the cellulose. So we need to understand what guides microtubules. So now, one of the behaviors of microtubules that you see is that what happens when they collide with each other. When a growing end of a microtubule hits another microtubule, it undergoes what is called a catastrophe. It's destroyed. Quickly, it gets destroyed. Well, the importance of that is that when you have lots of microtubules interacting, then when they hit each other, all right, they tend to get destroyed. They tend to kind of statistically, what happens, they tend to form alignments, because alignments, when microtubules are aligned, they tend to not hit each other so often, so you tend to get alignments just through this rule of hitting. So if you apply this rule, this collision rule, to a bunch of microtubules, and this is now a simulation or model. We've heard about simulations. If we let this run, what we see are these microtubules going all over our sphere, but it's not wrapping in any particular way, okay, because these alignments are forming essentially at random. So how do we go from this situation to a wrapping that I did with my balloon, all right? Well, suppose we introduce the notion of two poles. These poles, think of them as regions at the end of our cell where the microtubules have a higher probability of undergoing catastrophe. All right, so all we're going to say is that when a microtubule enters one of these regions, it's more likely to undergo catastrophe. Now, if we run this model, what happens is that microtubules get punished if they head towards the poles. And the result of that is that they tend to favor the other alignment. All right, so you now get them forming that alignment, and now your cell grows along an axis. All right, so we can see just by repetitive rules, these microtubule rules, these filaments, obeying these rules, we can end up with changing the shape of our cell. But that's now raised a new question, I hope, in your mind. What makes these poles different? And do we have any evidence that the poles are different? Well, in fact, we can label certain types of proteins in the cell, and that's an experiment shown here. We take our protoplast, we can identify certain proteins that tend to localize at single ends. Do you see that protein is going preferentially to one end of the cell? All right? So that shows you that there are indeed proteins that distinguish the two ends of the cell, and that essentially gives every cell a kind of polarity. It has two ends, all right, that are distinguished from the rest of the cell. I show this arrow or vector showing this direction of polarity. So now we have a new question. So we can imagine now that we have a cell that it has different proteins at its ends. These proteins can then influence the behavior of the microtubules. Now we have a new question. What controls the polarity? All right, so let's think about that. Well, fortunately, we're indebted to a mathematician, Alan Turing, for coming up with a model, an idea as to how polarity could establish itself. So Alan Turing is best known for his contribution to computers and computational theory. But Turing was also very interested in biology. He was interested in this problem of morphogenesis. In fact, he coined the term morphogenesis, which is all about the generation of form in plants and animals. Turing came up with an idea in which he said, suppose we have a molecule that promotes, this idea is called reaction diffusion. Suppose you have a molecule that promotes its own production, all right, a self-promoting molecule, a molecule that drives its own synthesis. What will happen? Well, this molecule will increase and increase and increase. Eventually, it would take over the world if this thing just carried on boosting itself. However, suppose this molecule also promotes something that inhibits itself. In other words, as it becomes higher, it also has an inhibitor that reduces the level of the molecule. And if we imagine that this inhibitor can diffuse away at a higher rate than the self-promoting molecule, then what he showed was that you can get patterns, interesting patterns emerging from that type of set of rules. So again, we're dealing with simple rules. From these simple rules, we can get a pattern. And that's illustrated with a simulation here, computer simulation. We have a kind of sort of noisy pattern to begin with. And then we apply Turing's rules. And gradually, all right, what happens is we end up with concentration focusing towards one end. All right, this is often called breaking symmetry, but I think breaking symmetry is a misleading term, because it's not that there's a point at which you say, aha, now the symmetry is broken. It's more of a continuous thing. And moreover, you'll see that this thing now has a symmetry still. We've just changed the symmetry. We've changed it to being more or less radial, or without having an axis, to one that now has a strong axis. So we can account then for the generation of polarity or differences through this reaction diffusion. So what have we learned from this? We've learned that we have a set of rules, simple rules that are repeated, okay, throughout our cell. And from these rules, we can see, if it's the reaction diffusion, we end up with a pattern. We end up with a pattern with with ends being different from each other. All right, that's the process of patterning. But then that pattern can influence the mechanics of the cell by changing the way microtubules are being organized, which are following their own rules. All right? And the microtubule then influences the wrapping of the cellulose, and so we end up with the cell growing this way. So what we've done is we've linking patterning, a pattern from Turing, to the mechanics, the physics of how the thing is growing. And I've shown it to you in a very simple setting, but fundamentally, that's what this illustrates, how you go from pattern to mechanics, how you connect those two, and how all of these depend on rules, repetitive rules. And the only reason these rules can be repetitive is because of the symmetry in physics. That is, the laws of physics are the same everywhere in the cell. If they were not, this would not be possible. All right? So the symmetry we're seeing, the symmetry change, absolutely is dependent on the fundamental symmetries of physics. Otherwise, we wouldn't be able to have these repetitive rules. All right? So that's essential. Now, I've talked about single cells, but what about more complicated things like tissues? Now, tissues are collections of cells, like a petal or a leaf. It's many cells together. So now let's look at a multicellular tissue. So this is a tissue in which we're seeing many cells, the outlines of many cells, but you'll see that each of these cells has a bright green bit that tends to be at one end. I don't know if you can see that. And that tells you, if you look at that bright green protein that's going to one end, it's because we marked it with a protein. It's the same protein that I showed you polarizing within the single cell. Do you remember there was a single cell where I showed you going to one end? It's the same protein. But now it's in all the cells. And it's always at one end of those cells. So what does that tell you? That tells you we have what I call a polarity field. We have every cell, it's like every cell has its own polarity. It has its own little compass. And all the polarities, all those arrows are pointing in the same direction. All right, so now we have a way of coordinating the growth of the entire tissue, because now every cell is coordinated with every other cell with regard to its polarity, and therefore it can change its microtubules and grow in the same way. So we have a way of coordinating the tissue. But then we've got a new question. How do we coordinate the polarity? What coordinates all these little compasses so they all point in the same way? Well, let's go back to Turing. But now let's apply exactly the same rules, but not at the cellular scale, but at the tissue scale. So now we've got a circular piece of tissue, not a single cell. I hope you can see that this is now made of lots of cells, all right? And we can run exactly the same rules, but at this higher scale. And if we do that, what we will end up with now, again, is this concentrating at one end. And if we now imagine that every cell also contains a little compass, a little polarity, then and suppose this compass is biased so that it tends to point down the concentration gradient of this molecule, then we'll end up with something like this. So now we've coordinated the polarity all between the cells, essentially with the same idea, but now at a different scale. Okay, so in fact, if we could have the lights up, there is a wonderful polarity field in this room. Can we have the lights on? Yeah, can anyone see, can anyone point to the polarity field in this room? No, they're pointing at me, pointing there. No, keep going, look around. No, they're pointing up. That's very interesting. Ah, somebody has seen it. Okay, look at that wall. So when I came into this room, I thought, my goodness, they knew I was coming. So there you see, so polarity fields have an aesthetic appeal, obviously. There all the arrows are pointing in the same direction. So, but the question is, now we have a polarity, how can that influence the growth of something as complicated, a tissue to produce these complicated shapes that we see in a flower? So now I want to do another experiment. Okay, what am I going to do? Yes. So what I have here is, this is not mysterious in any way. I have a piece of paper, and this is normal photocopying paper, or printing paper. Any one of you can do this experiment at home or anywhere you wish. The great thing about science is it has that anyone can verify and test whether a claim is true. So what I'm going to do is I've got three pieces of tape. And I'm going to take, first of all, I'm going to cut this one that's going across the top. I'm going to cut it off the piece of paper. And I'm going to take this and I'm going to put it in the water. So it's a very sophisticated experiment. Okay, so what's going to happen? Okay, I don't know whether you can see that on the camera. Can you see on the camera? Maybe you can zoom in. Okay, it's curled, all right? Does anybody know why? Can anyone tell me why it's curled? Yeah. Sorry? Heat it up. That's a very interesting idea. Thank you. Are there any other ideas as to why? Any other suggestions? Yeah, go on, another one. There's a what? Different size. Different size. Different size? Yeah. No, that's I think the correct. That's very good. So what's happening is that the water, the paper is absorbing the water. And when paper absorbs water, it expands. And because the paper is expanding, but the tape is not expanding, the tape gets bigger, but the tape does not want to get bigger. We have a conflict. The paper wants to be big, the tape doesn't want to be any bigger, and by curling helps resolve the conflict. Okay, so that now we've got a curled piece of paper. All right, so it's a conflict that's changing the shape. We're getting a curvature arising, or this type of curvature arising from a conflict between two things trying to do something different. Now I'm going to do this experiment. Now I'm going to take the other piece, all right, running along here. I want you to think about what's going to happen. Okay, I'm going to put that in the water. What's going to happen now? Okay, I hope this should work. Right, can you see what's going on? It's curling in a different way. Okay, do you see that? So, why? Why is it curling in the other way? What, sorry? Is somebody saying something? Sorry? It's not equal in both directions. Is that what you're saying? Yeah? All right, that's what we heard the word anisotropy. The paper is anisotropic. The fibers are actually oriented preferentially in one direction. This paper is made of cellulose fibers. These fibers are oriented pretty much in every piece of paper that you have. They're not randomly oriented. They tend to be oriented along the long axis to make the paper less bendy in that direction. All right? So again, it tells you that the anisotropy, if we have anisotropy, we can orient the shape change. all right, by controlling which way we orient our growth, in this case, the expansion of the paper. So now I'm going to do the final one, the diagonal one, and I want you to think about what's going to happen when I do the diagonal one. Any suggestions? What? Spiral, I heard. Let's see, we got predictions here, and we're doing experiments to test the predictions, which is always good. All right, so now let's see. There you go. Okay, thank you very much. So let's go back to the slides. We go back to the slides. Okay. So what I've shown you then is that we've now got various concepts of polarity fields, of conflicts between growth, and I want to briefly say how that will help us make this transition from the helical symmetry to the mutations through to the shape of the flower. All right, so now imagine if you look at, okay, let me say this. The cycloidea mutant, the mutant that I've showed you that has radial symmetry, is a mutation in a gene. Remember. Now this gene is actually called cycloidea. Biologists name the genes often after the mutant. And what does this gene do? So this gene is actually a gene that encodes a protein that switches on other genes. But why is it affecting the symmetry of the flower? Well, it's related to where this gene is active. So now let's go to a very early flower bud. A very early bud that will give rise to the flower looks like this. Like a loaf of bread. Looks like this, I mean. Like a loaf of bread. And you may see the individual cells on that loaf of bread. Now, at this point, you would see that the top of that loaf will give rise to the top of the flower, the bottom of the loaf gives rise to the bottom of the flower. All right, but at this stage, there's no obvious difference between top and bottom. All right, but if now I slice the loaf in half, and I look on the side, and I'll ask, where is my gene, this cycloidea gene, active? You see that it's only active in one part it's on the upper part. So that the dark blue color that you see, the dark blue color that you're seeing is where the gene is active. So it's a very exciting result that we got. We found, aha, the gene is only active in part of the flower. And you'll see that that's the sort of thing that we've been seeing with our reaction diffusion. It's being concentrated asymmetrically across the flower. Now let's combine that idea with reaction diffusion to see how we might generate asymmetry of the flower with different petal primordia arising around it. So here we're going to do another reaction diffusion at the tissue level. And what you'll see is, first of all, the first reaction diffusion is going to produce this asymmetry from top to bottom that relates to the cycloidea gene, so the equivalent. But then we'll introduce another reaction diffusion, shown in blue, and we're going to grow our ring. And if we have it such that the first one restricts the second one, then what happens is we end up with spots. This is all simply a result of this repetitive rule of Turing's rule. All right, we will end up with these spots arising around. Now you just have to imagine that these spots, so now we have bilateral symmetry. We have a difference between the top and the bottom. We have these regions that are going to form primordia, or the buds that will form the petals. So now imagine we take this idea, we elaborate it a bit more, and we imagine we've now got a flower bud with these different primordia that have grown out, but that have a difference between the red region and the rest, the upper region and the lower region. And you'll see there's arrows in our representation because these arrows represent the polarity field that's going to help orient the growth. But now what's going to happen is these genes are going to change these growth patterns so they come into conflict. And we're going to end up with deformations resulting from these tissue trying to grow in different ways. Now I'm going to magnify this so that you can see these conflicts and without changing the scale of the image. So what you can see then, if you put in some rules, and you put in these conflicts, you can arrange it so that you end up with a flower growing out. So nothing has told this flower to grow out. That is purely a result of the conflicts in growth patterns that were put in there. Now this is a model. This is a computational model. We don't know exactly all the different components that go into this model, but it shows you in principle how you can, through these conflicts, oriented by these polarity fields, get quite complicated transformations. Now let's imagine that our protein is missing, the one that distinguishes the top from the bottom. Now this just shows you it growing in, this just shows it to you growing in scale, all right? So all of this is happening as it's growing. It's not, okay? So now, yeah, let's go back and remove the asymmetry, the asymmetric gene, the one that's expressed at one end, and now we've got a symmetrical starting point. We apply the same rules, but now it's all following the same, each petal now is following the same rules, and if we grow that using the same principles, the same rules, we end up with a radially symmetrical flower. So what I've shown you then is that, very briefly, very quickly, is how, in principle, we can go from a mutation, we can change the symmetry of a flower because of these principles that link the patterning process and the mechanics, the formation of these polarity fields and the growth conflicts that lead to this transformation. But in doing so, I've had to bring in a whole series of rules. All right, and the question is, where do these rules come from? Okay. Let me just repeats that point. So we start in going through this these transformations. We're going through different scales, each of which exhibits symmetries. We start with the atomic scale, the molecules, the molecular scale, where we've seen molecules like microtubules and cellulose. We've gone through to cells, where we've seen wrappings in certain directions and polarity. Then we go to tissues with polarity fields. And at each of these levels, we see different symmetries. And in all these cases, we see that these symmetries are a reflection of rules, simple rules, applying at different scales. So you go from one scale to another scale. But as I said, where did these rules come from? And for that, we need to, we thank another great biologist, Charles Darwin, who really came up with an idea as to how the rules are played with through the process of evolution, through natural selection. And so I now want to say a little bit about natural selection and give you an illustration of how natural selection operates. And again, I'm going to use my favorite flower, the snapdragon antirrhinum. So it turns out that you can find two varieties. This growing in the wild in Spain, you find two varieties of this plant, one that has yellow flowers with a magenta highlight, all right, the one on the left, and another one that has magenta flowers with a yellow highlight, the flower on the right. And these are two ways that the flower signposts to bees. So a bee comes along, it needs to get into the flower to pollinate, and these provide alternative ways of signposting the bees. They're equivalent ways, but they're different ways. Now, these plants grow in Spain, and mostly the yellow ones grow in one area, and the magenta ones grow in another area. But sometimes they come into contact, and there's a road, this is a road in Spain, where you'll find yellows on one side and magentas on the other side, and then in the middle, where they come into contact, there's 1 kilometer, where you find them hybridizing and producing intermediate forms, different intermediate colors. So why is it now? We know that this contact is ancient. It's been hundreds, if not thousands of years old, this contact. So why have the colors not mixed? Why, over time, have these colors been maintained so that there's still only 1 kilometer where we find these colors? To answer that question, well, we thought maybe these hybrids produce less progeny, okay? That they're less fit, they're less attractive to the bees because maybe the colors are mixed together and don't work very well when you combine them in this way. So we wanted to test that idea. Now, a few years ago, you wouldn't have been able to test that idea very easily, but now we can sequence genomes. We can take an individual and sequence its DNA, and that allows us to do pedigrees. We can see who was the mother, who was the father of this individual by comparing their DNA. So what we did was we collected every year, we went along this road, we collected every individual, And then we looked at its DNA, and then we did it the next year and the next year, so we could figure out who was what progeny, how many progeny each individual left. All right, so it's like doing a big family tree, but not for humans. We're doing it for snapdragons. Okay, much more interesting. So what I'm going to show you is a fly through the valley. We're going to fly through the valley, and every pin that you'll see, the color relates to the color of the flower of that plant. All right, so here we go. So first of all, we're seeing magenta, all right, magenta pins. Now we see some hybrids, pinks, whites, orange. We're going into the hybrid zone where these are mixing together. We keep going, and now we're starting to see yellow. All right. So who said snapdragons were boring? Who said anti-rhydum was boring? So exciting. Right, so from this type of information, we can then compare every year. And you can draw lines connecting, seeing who's leaving, progeny, and so forth. And then from that, you can calculate what's called a fitness, what's called a fitness landscape. So each column here, the height of it tells you how many progeny individuals with different phenotypes, different colors were leaving. And you can see there's two columns that are much higher than the other two. They're the ones that have the parental color, but the hybrids have lower fitness, all right? So it fits, so it's a way of testing that idea and showing that indeed the fitness is operating in this way, and that selection will therefore act. But the important thing is that we're now dealing with populations of individuals, right? The natural selection is operating on a population of individuals. I showed you all those individuals. It's a phenomenon, it's a population phenomenon. All right, now we've really been applying simple rules now at the level of populations of individuals, the rule of natural selection. So if you think about it, we've been dealing with populations all day. I mean, this entire lecture. We've had populations of atoms, populations of molecules, populations of cells, populations of tissues, populations of organisms, all right, or individuals. And natural selection is a simple rule operating at the level of individuals that then feeds back to influence the genes at the molecular level. So that then the genes that are passed on will be influenced by natural selection, and so you go through this entire loop. And all of this depends on the symmetry. These rules operating each level depends on the symmetries of physics. If every individual obeyed different rules of physics, there's no way natural selection would work. All right? It only works because every individual obeys the same laws of physics, and so trends can emerge that favor, in general, one type of adaptation versus another. All right? So it's all based on the symmetries of physics. All right? But what we're seeing is these repetitive rules operating at all these different levels. Now, what I've said about, so this is just summarized here, just as in the water molecule, every atom follows the same physical rules, so every individual cell or molecule in a living organism follows the same physical rules. Now, this applies not just to flowers, it applies to every animal and plant, and it applies to us. But with us, there's an extra special twist, all right? Because not only are we the outcome of the rules, we're also the originators of the rules. We come up with these rules, okay? We discover the rules. So we're discovering the rules while being a product of the rules. Okay, that's kind of weird. And different people deal with that issue in different ways. I'm going to show you how I like to think about it. And it's shown by this picture by M.C. Escher. Now, if you look on the left of the picture, you see somebody in an art gallery. Okay, you see a picture of somebody in the art gallery. And what's the person doing? He's looking at the painting, a picture in the gallery. And if you follow the picture around, The picture brings you back to the person that's looking at it. So the person looking at it is actually part of the picture. And I think that represents the way we are, all right? The world in which we are, that actually we do see rules. We come up with rules, but we have to remember that we're also the product of the rules. And through that, we nevertheless can make sense of the world to a remarkable degree. But we have to remember the way we're looking at the world is to some extent going to be affected by our makeup, all right? There is at the deepest level an interdependence, which means that certain things might seem easy to us, other things might seem difficult to us as a result, because of the way we are constructed from those same rules that we're trying to discuss or we're trying to discover. So, in a sense, the symmetries are out there. They're part of the physical world. They're part of the biological world. They're also embedded within us, looking at those symmetries. And so, to my mind, symmetry is actually a wonderful way of unifying the fact that there are these simplicities that lie at all these different levels. How we look at them may vary, but I believe that deep down, symmetries are really telling us something very important about the simplicity and unity of the universe, including life. Thank you.
Thank you very much, Professor Coen, for this fascinating lecture. Biology is so different from physics, but yet you showed us how it can also follow symmetrical patterns, so thank you very much. Please take a seat.
Yep.
Je repasse brièvement sur le français pour inviter nos deux panélistes de ce soir. Il s'agit de Jean-Philippe Uzan, cosmologiste, directeur de recherche CNRS à l'Institut d'astrophysique de Paris, que vous avez pu entendre lundi sur le temps et sa construction en physique, ainsi que le professeur Edmund Copeland de l'Université de Nottingham du Royaume-Uni, que vous avez pu entendre hier sur l'histoire de l'univers. Please join us on stage, please. Thank you very much. Je vais repasser sur l'anglais pour ouvrir, comme d'habitude, comme toute la semaine. We're so lucky tonight to have both physicists, both physics and biology in the same room on stage. It's kind of a rare event. So I'd like to open up this Q&A session just asking you, Ed and Jean-Philippe, if you have any reaction, comment, question after Enrico's talk. Sorry.
Hello, is that on? Yeah? No shortage of questions. But wow. I mean, there's so many things came out that, but I'm sort of a bit nervous of suggesting them because I could be completely wrong, but you know, your polarity field looked like you know, something was making it going in a given direction and then leading to this polarity. Yeah. And that just reminds us of phase transitions in the early universe, which can create things called topological defects, where the fields will align in such a way that there'll be configurations, which on one side, you know, in this case, some energy density on one side, and then they're in the vacuum on the other side, and they can play a big role in cosmology. We don't know if they can, and I don't know if it's relevant, but it just reminded me the way you started it. You had all these cells, and within the cells you had these arrows, and that's exactly what you do. You put the phases of the field are like the arrows, and how they orientate then gives you these defects. Yeah. So yeah.
That's very amazing. Interesting. Very interesting.
If it's relevant, it might not be.
Well, I think it's relevant in the sense that I would guess your topological fields, again, these emerge from repeated application of simple rules, and that's the point. That these rules, in a sense, nature is kind of boring. It doesn't invent new things. It does invent new things, but it repeatedly uses the same thing again and again and again, and that's why we see these similarities, because they're repeating. We're seeing this repetitive nature. That's the way I would think about it.
Jean-Philippe?
I have so many questions because I think it's very...
Maybe pick one or two.
Okay, so one thing is that I had the feeling that what is happening here is that because of this retroaction, the way that some kind of more fundamental laws express themselves is due to the change of the environment in some way. Is it a correct way to look to understand these things?
I think, so yeah, the environment influences, as we've heard, the way the rules play out. And a good example is the snowflake. Okay, every snowflake is meant to be different. I'm not sure if that's true, but the reason that there's so much variation in snowflakes is because although every snowflake, all the arms of the snowflake are experiencing the same environment, the snowflake itself, as it's crystallizing, passing through the cloud, it travels through different environments. And that means that one snowflake will have a different history from another snowflake. And so the rules play out in a different way. And we call that's the environment of the snowflake. And I think the same is true in biology. Clearly, if the rules didn't play out in different ways, we would all look the same. Okay, I mean, we wouldn't look the same. The universe would just be nothingness, all right? It would just be uniform. So although the rules can be very simple and repetitive, they may nevertheless be tweaked and varied in different ways. And what you call environment and internal, of course, is also a question of scale. So you could say a cell inside a plant, the cells around it are its environment. Or you could say, no, you have an individual and the environment is outside the individual. It depends on how you define things, what you mean by the environment. But the general notion that, in a sense, given an environment and a content, then the rules can play out in different ways depending on the environment.
And then, if I can, there is one thing. Maybe I was very confused because, okay, I have to, which means that I have to think. So you had a genome, like a DNA. Then you talked about natural selections, and then you had this this dynamical system evolution. And so I was wondering, when you look at some kind of animals, it may have strips and spots and whatever. There are features that are coded in the DNA and can be selected over, and some part of other things that seems to happen due to maybe the size of the animal or the way the time scale with which it will grow, and that are not coded directly in the DNA.
Okay.
So the question is whether everything is, like, if you take the things about the leopard, I don't remember the stories, that the tails are strips and otherwise it has spots. Is it coded in the DNA or in another way and it expressed later on?
Well, I'm glad you're a little bit confused because I was super confused when I'm trying to think about general relativity and things like that. So that's that. I think there's a massive confusion over, not just in you, in terms of DNA and the organism, all right? So in a sense, genes don't do anything, all right? What genes are doing is influencing physical processes, all right? And they're doing it through affecting RNAs and proteins, and all these things are influenced. So there isn't a sense in which what is being done by the DNA and what is being done by the organism. The DNA itself doesn't do anything, in a sense. It's just there. The information is then copied into these other molecules, and they're the things that do things. So the spots on the animal and so forth, can be affected by the DNA, because if you mutate the DNA, that can change the pattern of the spots by changing a protein that's involved in making the spots. But the DNA is itself not producing the spot, all right? So it's always indirect, because the DNA is just an inert molecule in the middle of the cell. It's just this double helical thing sitting there. It relies completely on everything else to do everything. But now, when you talk about the spots and the stripes of an animal, when Turing first came up with his idea, the first application of reaction diffusion was to explain spots on animals and stripes on the zebra. But Turing said, talking about the zebra, he's meant to have said, the stripes are easy, what about the zebra? What about the animal? All right? Because that's what interested him, okay? How do you make the animal, not the decoration on the animal? And in a way, that's always been a real problem. How do you, that's the problem that we've been talking about. What I tried to show you today is how you can start to think about that problem. Because I gave you, for example, an illustration of how you could link the Turing pattern within a cell to then the mechanics of how that cell grows in a certain way. So it's bridging the notion between pattern, like a zebra stripe or a cheetah spot. You're bridging the notion between a pattern and mechanics, the mechanics of growth, the mechanics of formation. Now, the patterned elements in the mechanic are not spots, okay, because those are on the surface of the animal. They're the proteins, the sort of proteins that I've been talking about that influence the mechanics of growth.
Thank you very much. I was also wondering, do these reaction diffusion things that happen are also responsible for the uniqueness of the human fingerprint, for instance? Is it the same mechanism that is at play when we look at the human fingerprint, which is unique from one human to another?
You're asking, how is a human fingerprint produced? And I'm afraid I don't know.
I'm asking if it's kind of the same mechanism involving Turing's theorem or not.
It could be.
It could be.
I have no idea. I don't know. I don't know if anybody knows how, maybe somebody in here in the audience knows, but I don't know how a fingerprint, but I see what you're saying. There's a repetitive pattern it's both repetitive and unique. All right, but there is a repetitive component, and so you would imagine there is some sort of repetitive rule that nevertheless it relates to this environment thing that depends on certain other conditions that means that it goes in a different way according to the individual. But I don't know what that rule would be or how it works. So, sorry.
Thank you very much. Et on va prendre vos questions dans la salle et à distance. Je vais commencer par me tourner vers le professeur Rodriguez au premier rang pour savoir, est-ce qu'on a beaucoup de questions ce soir sur le...
On en a plein.
Peut-être 2 ou 3 pour commencer, peut-être.
Alors, je vais commencer. So I'll start with one which is quite general and that I guess many people are asking. Why is symmetry attractive for humans?
Yeah. Why is symmetry attractive to humans? Well, there is no single answer to that. But I would say, my answer would be that humans, like other animals, are attracted to patterns, okay? That we find patterns interesting. And why do we find patterns interesting? Well, we find patterns interesting because they help us predict, all right? I can predict that you're a human and not a plant because I see a different pattern from you than a plant. So we're looking always for patterns because patterns help us recognize things. And that's very important in allowing us to predict whether you're going to be, what you're going to do, what I should do in relation to you. So prediction is very important, and therefore we're recognizing patterns. And so one of the patterns that we recognize is symmetry, all right, because it's a very useful pattern, for example, to distinguish a rock from an animal or a plant. So I would say that's part of the reason that we're fascinated by symmetries, because they represent patterns. Now, the symmetries that we've been talking about in physics and biology are also patterns, and we get a thrill about we can get a mathematical pattern, or a mathematical symmetry or a physics symmetry. That can also excite us, again, because it allows us to see, through that pattern, we see a certain sort of an ability to predict or see an underlying simplicity. So I would say that it's to do with being able to predict and our natural interest in looking for patterns.
Thank you. Maybe I can add one other explanation or added explanation for that. Some people believe that symmetries in humans are used, at least when we look at other humans, as a proxy for fitness when we choose a mate. And that our general taste for symmetry would be a side effect of this pressure for finding fit mates.
Yeah, no, I don't think there's a single explanation. So, but having said that, I don't think you don't see people going around with highly asymmetric faces. Okay, I mean it can happen, unfortunately, and that's usually considered not a good place to be. But most of us are reproducing with people that actually have almost symmetric faces. So I'm not sure that there's very strong selection for symmetry. I'm not even sure that somebody that we find beautiful is going to necessarily be much more symmetric than someone. You mean you can be, I don't know what beauty and ugly are, but if we allow to use these terms, you can be ugly and symmetrical, and you can be beautiful and symmetrical. So I'm not sure how powerful that selection is. I'm not saying it doesn't exist. I wonder how powerful.
So maybe a second question. Which selective advantage does a radial versus a bilateral symmetry provide for a flower?
So it depends on the flower. So you take an antirrhinum, a snapdragon, radial symmetry is for sure disadvantageous. Because if you don't see that, the mutant that I showed you would not survive in the wild because a bee wouldn't be able to get into the flower. So that pollination mechanism, the mechanism by which it is pollinated depends on its asymmetry. But there are species that have more symmetrical flowers that haven't evolved this way of targeting pollinators. So in those cases, it's okay to be symmetrical. So there's a diversity of approaches. Some species adopt the symmetrical form, some the asymmetric form. I don't think we know really why one is favored. There's always a problem when you're comparing species to think about which is better than the other one. When I'm looking at a population, I can think about fitness. I can say this individual has higher fitness than that set of individuals because it's in the same population. So I can tell you in a snapdragon, it's bad news to be symmetrical, okay? Radially symmetrical, for sure. But if I was to now say, compare a snapdragon to a buttercup, which is better, a buttercup or a snapdragon? It kind of doesn't make sense. It's like saying, which is better, a lion or a cheetah? Do you see what I'm saying? So you're not comparing the same thing. You're comparing very different things. And so you can't really make that comparison. So both strategies work, for sure, in different species.
One very short question. Yep, sure.
How can microtubules be symmetric while being composed of non-symmetrical molecules?
So they're not. They show helical symmetry. All right. So they're not because of the way they're assembled. So in fact, asymmetric molecules, what are called chiral molecules, molecules that are handedness, often will assemble to form helices. All right. So if you take a handed molecule, something that's asymmetric, you put lots of those molecules together and you stick them together, you often get spirals because that asymmetry propagates. All right, so that way you end up with a spiral structure with a handedness, reflecting the handedness of its components. That's very different from what happens in diffusion, for example. If I take a handed molecule and I have lots of handed molecules and allow them to diffuse, they don't form a handed. Diffusion is not handed. It ignores the handedness. But if I assemble them physically together, then they can often adopt a handed conformation. In fact, that's more often the case. They'll often adopt a spiral form.
I have a quick question for you, Enrico. During our preparatory talk, you told me that at some point, people were tempted to make physics stand on its own without involving us humans in the equation. But maybe they shouldn't do so. You know, just erasing human from the picture when we're just trying to make the physics stand. So could you please elaborate a bit what you had in mind, and then we will ask your colleagues' reactions.
So it's a feature of science that the observer is removed from the observed. All right. If you look at a scientific paper, you take away you try and take away the personality of the individual. You try and make it all very objective. That the world is there. We're observers, and we're all very objective. We don't have emotions, and we observe what's out there. That's a useful way of doing science. It has a value because it means that everyone can do the same, can validate. So everyone, for example, can go and cut pieces of paper and check that what I've done is the same, and it doesn't depend on whether I do it or you do it. So in that sense, science is based on the notion of a separation between us and things that we study. However, when we look deeply into our relationship with the world, and we ask, what is reality, all right? Is reality independent of us, all right? Then we have to say, maybe, my view is, in a sense, you have to ask, is it useful to use the word reality? But even if you use the word reality, the way we look at reality, what we call reality, let's call it the thing that we are not, as it were, the thing that's out there. The way we look at what's out there depends on us, all right? But it doesn't mean that there are things that we're discovering that are features of what is out there. You see what I'm saying? So I can see simplicities, but the way I describe those simplicities depends on me. So, for example, let's take the rules of quantum mechanics. So the rules of quantum mechanics are very complicated for us to understand. Is that because the rules of quantum mechanics are more complicated than Newtonian mechanics? Or is it because we live in a world where we familiarize ourselves with Newtonian mechanics is based on more on our daily experience. If we lived in a world in which things behaved according to Heisenberg's uncertainty principle, maybe we would find quantum mechanics really easy and Newtonian mechanics very difficult. So it's the way we bring to the world certain things that we find easy and some things that we find difficult. But that doesn't mean there aren't beauties and symmetries out there. It just means we have to remember we're always looking at them through a particular lens, which is itself dependent on what is out there.
Thank you very much. Do you have any reaction, Ed or Jean-Philippe, to this?
No, I think this is going to what we were discussing this morning, because at the end, I told Enrico that Feynman used to say about quantum mechanics that you never understand, you just get used to it. And I think this is exactly it. I mean, we get used to some formalism, to some way of living in a mathematical world, and then we get intuition in this world. And each of us, with the same theory, we don't develop the same intuition. I'm sure, like, if Ed and I were starting to discuss cosmology or gravity on the blackboard, we start from the same equations, but we don't picture them in our mind in the same way. And this is why the discussion is fruitful, because on a given problem, you say, I see this way. No, I would attack the problem this way. And there is all this part of intuition that you need to assess at the beginning of a research project. And to me, there is a kind of artistic creativity in that at that time. And then, of course, you want to rationalize everything so that anyone can go to your result. But this is the second step. And usually all this creative part is put out of the scientific paper. And this is just when you are on stage like that, that you're allowed to tell a little bit how you make your discovery. I mean, when you make some.
No, I think that's right. That when you look at a scientific paper, if all you did was look at scientific papers, you get this impression that humans didn't write them almost, okay? But when you're doing science, it's an incredibly creative exercise. It's just that we hide that, in a sense, because we're trying to share things that are independent of where we're coming from. But I think the richness of science does come from the fact that we all think in slightly different ways. Yet, we also have a commonality that allows us to agree or disagree in a consensual way.
Thank you very much. Do you want to add something? On va prendre des questions du public. Donc, levez la main, on pourra vous donner le micro. Oui, monsieur, là-bas, à droite. Merci.
Thank you very much for the fascinating lecture. I'd like to go to your example that you had of the
could
five.
You please a bit louder?
Pardon.
Yes,
Is that easier? I'm sorry. Okay.
Perfect.
Thank you very much for a very fascinating lecture. I'd like to go to your example of the 5-petaled flower. And you illustrated it as a mutation, and then with your model, you showed some small tweak to some rules, which ended up with a 5-petaled structure. I'm just wondering, was that the first tweak that you made to your model, or did you do several tweaks? And did you sometimes have a 3-petaled structure, or a 7-petaled structure, or a 13-petaled structure? And if you did get structures with different numbers of petals at the end of your simulation, were they very minor tweaks to some of the parameters of your model, or were they in fact major inputs? I'm just sort of wondering if there are any reason why it was 5 and it wasn't 7, and why does the Antirrhinum have these mutants of 5 and not of 15?
Right, now that's a very good question. So There were two models I showed you. One was the one that grew to form the flower. But that one I started with 5 to begin with. So it doesn't answer your question in the sense that I put the 5 there, all right? I didn't ask the 5 to emerge. But the other model I showed you was a reaction diffusion model in which you saw those blue spots arising, which was kind of a representation of the idea of generating lots of petals. And that one, if I run it for longer, I will generate more spots, which is your point. Which shows that, in fact, you could generate a different number just depending on the strength of the inhibition and how much I grow it. But that model was kind of abstract. And the question is, in reality, in a growing bud, how is that controlled? How is the number controlled? I mean, I gave you a very simple reaction diffusion. It's not as simple as that, we know, because there are other things going on in terms of polarized transport of things. And that's a very interesting question. It's an open question. We don't know. I think maybe I should say, what I've tried to do today is give you a sense of how things could be. It's based on science, but we're very far from really understanding this to the level of what you're asking is exactly why is this happening versus that happening. We're getting some clues, we're beginning to understand, but we're still incredibly ignorant about many of the whys and wherefores.
Does it answer your question? Yes. Oui, madame, du coup, à gauche, un micro, s'il vous plaît.
Thank you for your talk. Do you find that clonal species are more likely to be asymmetrical because they're not influenced by pollinators? I mean, how can you explain why some species are not symmetrical and others are? In flowers, I mean.
Are you asking why are some species more symmetrical than other species? Say, in terms of flowers, or
Yes.
Is that what you're asking?
Yeah.
Yeah. So, as I said, we're not entirely sure. We know both strategies work. Okay, so an antirrhinum and an orchid will have bilateral symmetry. Okay. But a buttercup or a tulip will have more radial symmetry, more planes of symmetry. So you can ask why one and why not the other? Yes? I think that's your question. And I think it relates to the fact that in biology, we find many different ways of surviving and reproducing. Okay? That's why we have so many species. Okay, there are in a given environment, there's lots of ways of solving the problem of reproduction and survival. If there was only one way of doing it, then we would all look exactly the same. There would only be one species. But there are lots of different ways, all of which are very effective. And then you have to ask the question, yeah, but why is this one used here and that one used there? And then I think it's really problematic because it's not easy to compare. All right, you just see both work. All right, maybe one will go extinct, maybe one won't. But let's assume they're both surviving. All right, it's difficult to say. We can only say that they both work. You can also ask the evolutionary question: how did their ancestor? They would have had a common ancestor. Why did the common ancestor, in this case, go? How did it become asymmetric in one case and more symmetrical in the other case? Usually, the ancestor is more symmetrical, and asymmetry evolves from symmetry, from greater symmetry. But I don't think that's a rigid rule. I'm sure you could go the other way as well. So I'm not really I'm not answering your question in a satisfactory way, because I don't think there is a satisfactory answer as to why, in evolution, different strategies are adopted. All we know is that evolution leads to many different strategies. And if you see, for example, if I compare a tulip to a snapdragon, it doesn't just differ in flower symmetry, it differs in all sorts of other ways. All right, so how do I know what the contribution is to the symmetry compared to all these other differences? I haven't got a controlled experiment. Whereas within the snapdragon, I can do the experiment, I can just change the gene, and it's just a single change. If I'm comparing two species, there are thousands and thousands of changes, and that's why it's so difficult to answer your question.
Est-ce qu'on a encore une question dans le public? Oui, tout à fait, premier rang. Deuxième rang, pardon. Voilà, exactement.
Mais les symétries dans la biodiversité, ça sert à quoi?
Symmetries in biodiversity.
Sorry?
I'm just translating.
Yeah. Sorry?
Symmetries in biodiversity, in animals. What does it? What's the purpose? What's the use?
What's the use of diversity? That's a very, very good question, and it's a very difficult question. Thank you. What is the use of diversity? I think it depends on what your how do I answer that? It's like saying, in a way, what is the use of physics, or what is the use of chemistry? There are things that just happen, and we're very grateful that they happen. So we're very, very happy that there is diversity, because you would not be asking me this question if you were a frog, okay? So we're very happy that there's diversity, because there are lots of different ways of solving the problem of how to survive and produce babies. A frog does it one way, humans do it another way, a flower does it another way. So there are many different ways of this happening, and each maybe has certain advantages compared to the other, which allows all of them to live together. But if you ask, what's the point of that? In a sense, I can't answer you. In a sense, I'm just saying that is the way the world is. All right? It's not that there is somebody saying, this, you should do it this way because it's better. It's just the nature of the world in which we live. Does that answer your question? I hope it does. Otherwise, I'm in deep trouble.
Thank you very much.
Sorry? It was a different question. Did I answer the wrong question?
No, just...
Okay.
It's just rephrasing the question.
What's the role of symmetry in biodiversity? Do you mean, okay, so, gosh.
Just answered it.
Another great question. It depends what type of symmetry we're talking about. So, in one sense, if we're talking about the symmetry, fundamental symmetries of the behaviors of things, all right, if it wasn't for those symmetries, we wouldn't have biodiversity, we wouldn't have individuals, we wouldn't have organisms. Because what I tried to show is that the evolution of animals and plants depends on symmetries. Without those symmetries, there would be no fundamental symmetries, is what I'm saying, these repetitive rules. Without that, there would be no, we would not be here. Actually, deep down, without that, the universe would not be here. All right, so in that sense, symmetry is essential to the universe, it's essential to life, it's essential to everything. Then there are other types of symmetry that we can ask, what is the contribution of flower symmetry, to flower diversity and so forth, which is another type of question, but I'll stick with the first. All right, that fundamentally, without symmetries, we would not be here. Okay, so because it's so important in terms of providing simplicity that underlies the production of all this diversity.
Thank you very much.
Does that answer your question?
Thank you very much for this very detailed answer in 2 parts. Thank you very much. I think it's 8 p.m., past 8 p.m. Well, we have to wrap up. I had so many more questions, but I have to end it. So thank you very much again. Please make a huge round of applause for our speakers and our panelists. Et voilà, vous le savez, je vais repasser sur le français quelques instants pour vous rappeler que même si le colloque Wright se termine ce soir pour les conférences, on a encore le spectacle Son et Lumière qui se tient jusqu'au 17 novembre au Musée d'Art et d'Histoire, tous les soirs à 18h30, 19h30, 20h30. Ça dure 20 minutes, c'est gratuit jusqu'au 17 novembre, n'hésitez pas. Et restez encore assis quelques instants, on n'a pas tout à fait fini. Je voulais encore remercier, moi, personnellement, nos orateurs et oratrices de cette semaine. Franchement, vos conférences ont été d'une clarté incroyable sur des sujets tellement ardus, donc merci beaucoup. Merci également à Thierry Courvoisier, président de la Fondation Wright, et à Fanensis Ban de l'Université de Genève pour leur confiance. Merci à nos interprètes qui sont là-haut pour la traduction en direct. Merci à notre équipe de techniciens en régie. Et bien sûr, un immense merci à vous, public, d'être venus ou de nous avoir écoutés en ligne. On espère que vous avez pu apprendre beaucoup de choses. Et avant de conclure, j'aimerais inviter le président de la Fondation Wright, Thierry Courvoisier, à dire un petit mot de conclusion.
Pour clore cet exercice, j'aimerais nous rappeler qu'il est bon de consacrer quelque effort pour enrichir sa culture en sciences fondamentales par les temps perturbés qui sont les nôtres. Donner du temps à la culture, à la musique, à la science, à la navigation aussi, est une excellente manière de prendre quelques distances avec des nouvelles difficiles et avec des soucis quotidiens ou sociétaux. Ceci participe à rétablir un peu de sérénité dans la société, une démarche salutaire pour nous tous ici, mais aussi pour ceux qui sont dans notre entourage, proche ou plus lointain. Merci donc à nos orateurs de nous avoir offert cette occasion de retrouver un peu de sérénité dans une réflexion un petit peu plus profonde que les premières pages de nos journaux. Merci aussi, comme l'a dit Sarah, à tous ceux qui ont rendu cette semaine possible. Ce sont mes collègues qui ont présenté nos orateurs aussi, Tess qui a géré les questions, les interprètes, les personnes que vous avez vues partout. C'est grâce à tout ce monde qu'on a pu passer un ensemble de soirées, ma foi, fort agréables. Alors à tous, je donne rendez-vous dans 2 ans. Et je me réjouis de vous y retrouver. Ouf, je ne sais pas si ce sera moi, tiens. Mais enfin, quelqu'un sera ici à ce moment-là. Bonne soirée, bonne fin d'année.